It's my pleasure to be sitting here today with Dr. Stephen Tregay. Dr. Tregay is the CEO of Forma Therapeutics and is also a Davidson alumnus, so it's a pleasure to have him on campus. And I'm going to ask him a few questions that are relevant to the particular recent topics in the course. And I'm going to open with Dr. Tregay, if you would just give us an introduction into how you became CEO of Forma Therapeutics and your career path that led you there.
Well, it's a great honor to be here and talking to everyone today. So my background was I was a chemistry undergraduate, went to Harvard after that to get my PhD in organic chemistry. Really found myself really being most interested in kind of how business and science interfaced. And so I had a chance to start doing business development at a company called Array Biopharma, and then had a chance to move to Boston, working for Novartis, a large pharmaceutical company focused on strategic alliances and how to build kind of a new effort in research that Novartis was embarking on. That then translated into a role in the venture arm of Novartis called Novartis Venture Funds. And that's where I got Forma going. And it really was an opportunity to think about what really was innovation in life sciences. So it's been an exciting opportunity to be part of.
So could you tell us a little bit about CEOs in general of biotech companies? Can you put CEOs in boxes? Do they all have PhDs? Do they have typically MBAs? Are there MDs? How would you categorize?
Yeah, CEO is kind of an interesting role in that you see all types coming in. You see people with business backgrounds, operational backgrounds, MDs, PhDs like myself. I think what really separates CEOs of early stage companies in particular is really just a desire and excitement around entrepreneurship. You really have to be passionate about what you're doing. You have to be really fully committed to what you're trying to build. And really that can come in lots of forms. I think the other important thing to realize as a CEO is you're never going to have all of the answers. So while you may have strength in finance or business or science, you have to surround yourself with great individuals. And really that's the number one role of the CEO, is to build a great team, make sure they're aligned and motivated and empowered to be able to deliver on that vision. And ultimately, that's what your role is, and the better people you can attract, the more successful your company is going to be.
So can you talk a little bit day to day? How would you work with the team that you have assembled?
Yeah, so, really what I view my role at Forma and most CEOs' role is really to be a spokesperson for that company, to make sure that the people internally understand the vision and where we're going, that the board of directors, which really governs the company, understands why we're doing what we're doing, understands where we're trying to go. And then externally, what are the types of partnerships we want? What's the public face of the company that we want? And so you spend a lot of time working with different groups, whether they be your direct reports or people on the outside, to really align with what that broader vision is. And I think the bigger the goal and the bigger the vision of what you want, the more effort you have to put into making sure everyone understands that clearly in terms of what you're trying to do and how to measure that. Are you on track to delivering on that? So it's a fun role. It's a chance to really talk about what you want. It's something where your excitement, your enthusiasm for the company really, I think, is key to how you position your organization.
Could you talk a little bit about Forma, what
Sure.
the business model of Forma?
Yeah, so Forma is a company that's about 6 years old, and it was really founded not on a particular technology or a particular area of biology, but rather really a study in how the world of drug discovery was changing. Genomic medicine, that people have heard a lot about, was really coming into its own. This was really changing how we thought about drug discovery, and so Forma was really a concerted effort to get at a new drug discovery paradigm, to work in target families, working in parallel to change the drug discovery paradigm from a very linear process to one of execution in parallel and leveraging both genomic information but also new emerging target families into an integrated drug discovery process to really prioritize creating transformative cancer therapies out the other end.
So when you discuss targets, the edX course is really designed around target-based drug discovery, but we're aware in the course that there is a little bit of a divide between target and phenotype-based discovery. Can you talk about how that affects, or if it affects, Forma?
Yeah, sure. So when you think about the drug discovery process, one of the challenges that you have is you can be very reductionist, and that's kind of the reduction down to a particular target. And that opens up lots of tools like computational technology, structure-guided drug discovery, because you intimately know the molecular target you're looking to drug. The challenge then becomes, how do you understand that role of that target in a much more complicated cellular environment? So phenotypic screening, you understand the phenotype, i.e. what happens to a cell in that complicated environment when you apply a drug to that cell, but you don't understand the target. So what Forma tries to do is really balance both of those. We do classically target-based drug discovery, where we use all of those tools, high throughput screening, X-ray crystallography, computational approaches, but we try to be very quick to understanding how that target works in a broader context. What is the patient population you want to go after? What is the disease setting you want to understand? Because at the end of the day, if you follow a very linear path, you're often kind of seduced into biology that may or may not be pertinent to the disease state you want. If you're going phenotypically, you may have a direct linkage to where you want to go from a drug, right? Because you understand the phenotype you're looking for. But the challenge is the drug discovery process may overwhelm you and you may never get to there. So we try to take the elements of both and really bring in the elements of both of those into the drug discovery process.
Okay, fantastic. Now, something you hear a lot about today is personalized medicine, and that's a term that I encounter some on the Forma website. Can you talk about
Sure.
personalized medicine and Forma?
Yeah, and I think personalized medicine has had a lot of misnomers. It's had a lot of promise. The way we really view personalized medicine is the ability to look and understand the genetics of disease. And let's focus on cancer as an example here. Classically, cancer has been, is it breast cancer, is it lung cancer, et cetera. It's really kind of origin, tissue of origin. But what genetics has pointed to, and things like the Cancer Genome Atlas Project and others, really point to it's a subset of each of those cancer types. Each has a genetic background, if you will, that is what is driving that cancer. And so it's no longer, and I'll bring up an example of Gleevec, which is really, I think, one of the ones that ushered in this era of personalized medicine. Prior to things like Gleevec, we classically treated cancer with cytotoxics, and what that means is basically poisons that kill normal cells and cancerous cells, but cancerous cells are growing a little bit faster, so they kill them differentially. That's why you lose your hair, you have terrible nausea. It's a grueling regimen to go through. Targeted therapies really start thinking about targeting what is truly unique about that cancer. So coming back to the Gleevec example, which was discovered by Novartis, a company that I'd worked at, they understood that a disease called CML, or chronic myelogenous leukemia, was driven by a genetic change, in this case what was called a translocation, to create a signal that was always on. So picture a light switch that's always switched on. That drove this cancer. But that was unique to those cells as opposed to normal cells. The target of Gleevec turns that light switch off. And in doing so, you have a safe drug that has to be taken every day because you have to constantly turn that light switch off, but is one that changed the disease from basically a death sentence to something where you had over a 90% survival after 5 years. So really turning cancer into a chronic disease with a side effect profile that would be no different than a cholesterol-lowering drug or some other drug you may have. So that's really the promise of personalized medicine, is understanding what is driving your cancer and addressing that. What Forma does in that space is we really view that as a way to focus our clinical efforts. Do we know that the patient population we want to go after? Because many of these alterations may only be 5% of lung cancer or 5% of breast cancer. We need to understand how to pick those patients. So like Gleevec had essentially 100% response rate in its early clinical trials, as opposed to maybe 5 or 10% response rate. Well, it's not surprising if only 5% of the cancer is driven by that biology, you'd see a weak response. We want to pick those patients and then understand how to select them for clinical trials and then bring a drug to market. And that really profoundly changes the drug discovery paradigm and the cost structure of clinical development.
So as you talk about selecting patients, is that a challenge for clinical trials, identifying, so you'll need to do some analysis on each individual patient to make sure?
Yeah, absolutely. So, and this is where genomics has really changed. So when the first genome effort was launched with the NIH in 2000, it was $3 billion in 10 years to do the first genome. But in 2008, really we had a real breakthrough in what was called next generation sequencing. And this has really dropped the cost of sequencing dramatically. And so actually just this year, a company called Illumina has announced that they can sequence the entire genome for $1,000. So we've gone from discrete tests, where you might test to see if you're HER2 positive or EGFR mutated, a discrete test to test one hypothesis as to whether you'd respond to this drug, kind of a classical companion diagnostic, if you've heard that term, to now being able to really mine very deeply a lot of genetic alterations. And so it's that massively exploding knowledge base that Forma really tries to capture in on, is to understand these molecular bases of cancer and then say that's where we think we should be investing our research dollars, because we understand how to correlate a target to a phenotype, whether it be in patient samples or cell lines or animal models, to now the clinical trials and the ultimate, the commercial application. And that's what we want to see, a real clear correlation of what we're investing in to what we would be testing in that hypothesis down the road.
So as you identify patients who are maybe going to more likely respond to a medication, it also creates opportunities because there are patient populations who won't respond, and then that's another opportunity for potential a different drug development.
Absolutely, and it's an important thing to recognize is that in understanding the genetics, being on the wrong drug in cancer actually can be a huge impact on the healthcare system. These drugs are expensive, but more importantly for the patient, if you're on the wrong drug for 6 weeks, that might have been the window where you really could have had a robust response. And by the time you get on the right drug 2 or 3 cycles later, if you're just trying this random approach, your cancer may have evolved to a point where it no longer would respond to that therapy. So there's a real need to understanding this if we're going to have a big impact on cancer. Cancer is a challenging disease. It's a disease that's not stable. Your treatment of your cancer in early stage is very different than the genetics of your late stage cancer. So getting it early, getting you on the right drug is your best shot to getting that durable response.
So Forma is also is not just focused on oncology. That seems to be where most of the work is going on in your company. How about other therapeutic areas that?
Sure. So one of the things that because we work in target families and we think about understanding the commonalities across related enzymes and related target families, what happens in that situation is we go in and think about where we might go in oncology, obviously a very robust area for this type of approach, but also the biology could take us in a lot of different directions. And so what we look for in commonality is large unmet medical need. And so cancer, obviously, it's quite obvious, but when you think about rare diseases, for example, are really an area that shares a lot of similarities. It's typically a genetically defined disease that has a root cause, often very high unmet medical need. Often it's impacting children who might only live to, at best 10 to 12 years old. Profound effect on the families and on the children. And we can understand the genetics behind that. We can understand how a drug intervention may really reverse that phenotype. And so we are looking for this high unmet medical need. I think the drug industry historically had really looked at incremental innovation. Lipitor was the 6th drug to market in a class of cholesterol-lowering agents. The society today can't afford that kind of incremental innovation. It really needs profound innovation. And so we look to other areas where we feel we can make a big impact based on where our science will take us.
Excellent. Well, Dr. Tregay, I want to thank you for making yourself available for this interview, for teaching us, sharing a little bit about yourself and teaching us about Forma, as well as just exposing some of the thinking in the biotech industry about different targets, personalized medicine, genomics, those sorts of topics.
Oh, my pleasure. Thank you very much for having me today.
Great, thank you.